Welcome to BioWise with Aanchal. Thank you for all your love and support that you are showing to this channel by subscribing the same. Those who have not subscribed the channel, kindly subscribe, and then we'll proceed with the chapter Cell. Part 1 and Part 2 of the chapter is already there, and the link of the video is there in the description. Those who have not seen the video, you are requested to go through the same. Today I am going to present you the third part of the chapter Cell, where we will be talking about the important diagrams, questions, and what sort of questions can be asked in the examination. So let us begin. So the chapter we are talking about is Cell. Cell is an important chapter for standard 9. It is chapter 2 in standard 9 Selena, and it is chapter 1 in standard 10 Selena. Though this chapter is not important for your board examination as such, because no questions are asked from this chapter, but the dependency of this chapter in understanding the other chapters is very important. So let us quickly study about cell, the important diagrams that can be asked in this chapter, that is important diagrams. Okay, we will be talking about which diagrams? The first one is the mitochondria. Mitochondria is important. Chloroplast, nerve cells, and the blood cells. These cells diagram you all must know when you know this chapter. Moreover, in this chapter we have studied about prokaryotic and eukaryotic cells. So diagram of the prokaryotic cell, that is a bacterial cell, you can see the previous video. The diagram is well explained with the differences between prokaryotes and eukaryotes. Then what is the difference between a plant cell and an animal cell? The unique organelles present in these cells are very important. Now in your textbook in standard 9 Selena, table 2.1, there is a table which tells you about the different organelles present in the cell, their main characteristics and their specific functions. Now every organelle can be asked in your examination for around 2 marks. So 2 functions are very important. Now children, you must understand when you are talking about the functions, functions generally talks about the verb. We cannot say the function of mitochondria as the powerhouse of the cell. The function of mitochondria is to synthesize ATP. Ribosomes, protein factory. It is another name given to ribosomes. What is the function of ribosomes? Synthesis of protein or protein synthesis. Is that clear? Similarly, when we say what is the function of lysosomes, they are the suicidal bags. What suicidal bags? They help in intracellular digestion. So we have to be very specific when we write the functions of the organelles. Is that clear? So let us begin with the diagram. I will show you a very easy method to draw the diagram of each of these organelles and the part of obviously the cells. So let me quickly rub the board. So the first diagram is of mitochondria. So let us quickly draw mitochondria. Singular mitochondrion. For mitochondria, you have to draw an oval structure. Please see how the drawing is going on. Stick to the margin since it is a two-layer. It has a two-layer membrane. So just stick to the margin. Draw the grooves. The inner membrane of the mitochondria is thrown into folds to increase the surface area for the ATP synthesis. Okay. You can slow down the video and see how the diagram is being done. It's really easy to draw. Okay. Now you have to, it has its own DNA. Right. You now label it. It's a outer membrane. What is the inner membrane of mitochondria called? It is called cristae. Okay. Then you have the liquid part that is present, that is called the matrix. Okay, and here it has its own DNA. This is your diagram. For diagram you have 1 mark and for the label you have 2 marks. So 2 correct labels you get 2 marks. Is this clear? You can take a screenshot of the diagram. outer membrane, inner membrane. The main function of inner membrane is this is the site of ATP synthesis. Site of ATP synthesis. Okay. And the matrix, here you have the Krebs cycle. Krebs cycle. Those who are in standard 9, we will study chapter 7, that is respiration in plants, and chapter 14, that is respiration in human beings. So there we will be talking about glycolysis, Krebs cycle, electron transport chain. So here ATP synthesis, because ETC, electron transport chain, happens in the cristae. Those who have seen the first and the second video, you will be aware what I am talking about, right? So this is what the mitochondria is all about. Let me quickly rub it and go to the chloroplast. Next we will talk about chloroplast. You all know chloroplast is a unique organelle to the plant cells because it helps in photosynthesis because of the presence of a pigment called chlorophyll, which is unique to plants. That is why plants are called producers, because plant can only fix the solar energy into chemical energy, which forms the foundation for all the living organisms on earth, or you can say which is the beginning for all the food chains present on earth. Okay, so what is chloroplast? A chloroplast diagram, if you see, it's the structure is this way, and it is also a double membrane structure. Okay, it has got coin like structures which are called thylakoids. Stack of thylakoids are called grana. Granum is singular, grana is plural. So you see how I am making it, children. It's very easy. You can finish the drawing in 2 minutes. Not even 2 minutes. Okay, so you have got the granum, and they are connected by frets, tube like structure which are called frets. Okay. These are called connected by frets. Now to label them, you have outer membrane here also. The inner membrane, you have got frets. The entire stack is called granum. Okay, and single unit is called a thylakoid. Okay, and the liquid portion here is called stroma. Stoma is a singular of stomata, and stroma is the fluid present or matrix present in the chloroplast. Now chloroplast is a site of photosynthesis. So where is chlorophyll present? Chlorophyll is present in the walls of thylakoid. So the light reaction happens in the thylakoid. And the dark reaction, that is fixation of carbon dioxide into glucose, happens in the stroma. Okay, dark and light reaction you study in the chapter 6, that is photosynthesis, in standard 10. Okay, in standard 9 you don't have anything related to the dark and the light reaction, but it is there in 10 standard. So understanding this organelle is very important. Okay, this is one kind of plastid which contains chlorophyll as a pigment, green color pigment, and is called chloroplast. Any doubt here? No? Take a screenshot if you want. Let me rub the board clear. The next diagram we will talk about is the nerve cells. The nerve cell again is important for your 9th standard, as the diagram can be asked in the examination, and at the same time the same diagram is there for your 10th standard also in the chapter nervous system. Okay, so let me quickly draw the diagram for you. Just follow how I do it. These are the axon terminals. Okay, there is a nucleus, there are granules kind of things present in the cytoplasm. Then there are dendrites. The granules present in the cytoplasm is called the Nissls granules. These are all dendrites. Dendron is singular, dendrites are plural. Okay, they are the receiving ends. They receive the impulse, the dendrons, pass it to the cyton, and then it moves to the axon. And then you have got the myelin sheath. Myelin sheath helps in insulation. So, it helps in the fast movement of impulse in the nerve cells. Okay, these are called myelin sheath. It is made up of Schwann cells. Schwann cells. So, this is your diagram. For diagram, you get 1 mark. Fine. Now, let me label it. This is called the dendrites. These all structures are called dendrites. Then these all granules are called Nissls granule. This is neurolemma. This is the Schwann cells. This is called the myelin sheath. Myelin sheath. This is the axon terminal. This gap is called the node of Ranvier. Okay, node of Ranvier, mind the spelling. Okay, and this part, the head part, is called cyton, also called perikaryon, and this part is called the axon. Is this clear, the diagram? For the label, that is correct label, you get 2 marks. So, any 2 correct labels, but I will say that you should write at least 4 labels in all the diagrams, right? Now, let me quickly rub the board. Next, we talk about the blood cells. Blood cells are important again. You have to know, identify which blood cells, what type of WBC is present. So, it is important to know the structure of the blood cells. So, let me quickly go through the blood cells. Okay, now when you talk about the blood cells, there are 3 types of blood cells. Okay, you have erythrocytes, you have leukocytes, and you have thrombocytes. Thrombocytes are called platelets, and leukocytes are WBCs, and these are RBCs. Okay, you all know that RBC is an oval spherical cell which can be this shape or this shape, that is concave, biconcave cells. They do not have no, that is, they do not have nucleus, no mitochondria, and no endoplasmic reticulum. I hope the reason is clear. No nucleus, so that it can accommodate more oxygen, no mitochondria, so that it could not, or it to avoid the usage of oxygen it carries, and no endoplasmic reticulum, so that it can change its shape and pass easily through the capillaries. Now, when you talk about the WBCs are basically of 2 types: granulocytes and agranulocytes. Okay. Now, when you talk about granulocytes, that means they have granules present in them. So, they are of 3 types. What are those? One is with a numerous lobed nucleus and dotted cytoplasm, that is granulated cytoplasm, that is why granulocytes. This is called neutrophils. Okay. Then you have E for ear, and we use headphone on our ears. So, you have a headphone shaped nucleus and a granulated cytoplasm. So, this is eosinophils. Eosinophils. Okay. And then you have bilobed nucleus. Bilobed nucleus, that is granulated, and this is basophils. Okay, is this clear? Now, agranulocytes, when you talk about, there are 2. One is lymphocyte, L for lymphocyte, L for large nucleus. So, you have lymphocyte and it has got a large nucleus, and the cytoplasm is not granulated because it comes under agranulocytes. And when you talk about the next one, that is monocytes. Monocytes, when you talk about, monocytes are the largest of the WBCs, and it has got a kidney shaped nucleus. So, this is your blood cells. You need them in chapter 17, that is aids to health, in standard 9, and chapter 8, that is circulatory system, in your standard 10. So, this is all about the blood cells. Kindly know the structure. This can be easily asked. How can you identify neutrophils, basophils and eosinophils? So, you can see the nucleus and you can identify the cells. Even lymphocyte, yes, L for lymphocyte and L for large nucleus, and monocytes have got a small kidney shaped nucleus, but the cells are the largest. Is that clear? Any doubt? But we can take a screenshot. This is all that is about the important diagrams that can be asked in this chapter. Moreover, in this chapter, we generally ask about the functions, some reasoning questions, as we have already seen about RBC. So, some reasoning questions can also come in your examination. Kindly by heart the table. At least you should know 2 functions of each organelle and why the organelle is given certain name. Why is mitochondria the powerhouse of the cell? Why is chloroplast the kitchen of the cell? Why is lysosome the suicidal bag? Right? Why is ribosomes the protein factory? So, you have to know the functions of each and every organelle present in a cell. At the same time, you should know which organelles are unique to the plant cells and the animal cells. So, with this, we conclude the entire chapter cell for standard 9 as well as for standard 10. Okay. So, any questions if you have, put it in the comment section. If you want any explanation which I have not given in this chapter and you want to understand it again, kindly let me know. You will be getting a reply for the query. Like, share and subscribe if you think it is worth sharing, and happy learning children. Thank you.